Hello and welcome everybody. My name is Rasouliyan. I'm the head of Biostatistics and Data Science at OmniHealth, and I'm here today with my colleague Kumar, who is a senior data scientist at OmniHealth. And today we're going to be discussing adverse drug event detection using Spark NLP. Just to give you an idea of how the talk is going to be structured, it'll be divided roughly into half. In the first half, I'll cover just the background of adverse drug events and go into a little bit of the epidemiology. And that will set the stage for Vic to get into the technical details of using Spark NLP for detecting adverse drug events. And so that's how the talk will be set up. And before I get into my section, I really just wanted to say a little bit about what Vic and I do, because I really think that will really help us understand the motivation behind this talk and kind of put it all into a lot more context. So Vic and I are with OmniHealth, and we are a healthcare data company, and we work with health systems around the country. We partner with them around the United States, and we work with their electronic health record data. So we go in, we de-identify their electronic health record data, we tokenize it, and we normalize it or standardize it into a common data model to where the data are completely, you can completely translate them across the different partners within our platform. And one thing that we're seeing more and more becoming very prevalent is the clinical notes in the data. And I think clinical notes are, as we know, they're basically unstructured text fields that a clinician or a physician might write about the patient for a particular characteristic or at a particular visit or something along those lines. And those notes are becoming more and more important. There's a lot of valuable information in those notes. And to give you an example, one use case that we're seeing over and over is the whole concept of pharmacoepidemiology. So, for instance, a pharma company might have a particular drug that's out there. It's gone through clinical trials. It did well in the clinical trials. And the regulatory authorities say that, your drug is approved to be out on the market. But, we found this one patient that got this really rare form of cancer, let's say, in your trial that was exposed to your drug. We want to make sure it's not a real thing. So they'll kind of say, you also need to do this long-term follow-up study where we can assess the safety of your drug in the long term. And so we want to see if this really is a thing associated with your drug or not. And so a lot of times these adverse events or these kind of bad things that may be happening from the therapy are actually collected in the clinical notes, and that might be the only place they're collected. And so these pharma companies, they do turn to things like electronic health records and real world data sources, claims and things like that. And so that's really is kind of the motivation behind this talk and why we think it's a super interesting topic to really be able to detect these adverse drug events in the clinical notes of EHR data. So a little bit of terminology and just some terms, because I think that a lot of times people use these terms interchangeably, and they're often confused with one another, but they really are distinct things. And so I just want to clarify a few things. First is adverse event. And so this is kind of like the entire universe of any unwanted or unfavorable occurrence that's temporarily associated with the use of a medical or investigational product. And so within this definition, there are a few terms that I want to call out even to give even more clarity. So when we say occurrence, that really could be anything. It can be a sign or a symptom. It can be an abnormal lab test, full-blown disease, or even death. So it's very, very much a wide net that's cast. So it's really kind of a thing that occurs after the use of a medical or investigational product. When we say medical or investigational, that can also mean many things. It can be a drug, a biologic, a medical device, or a combination thereof, right? So it may not necessarily refer to one specific entity. And then finally, the term associated does not necessarily imply causation. So we're not necessarily saying that the event happened because of the drug. We're not implying that causal relationship between the two. And I think that's a very distinct thing that we need to keep into mind when somebody uses the term adverse event. On the other hand, when we say adverse drug event, that the distinction there is that causality is implied, or at least it's suspected. So it's an adverse event related to the drug. So you can think of an adverse drug event as kind of a subset of an adverse event where the event is related to the drug in some kind of causal manner. It's also important to note that it can include medical errors and overdoses as well as the expected responses, right? So there is an element of human error. So in a situation, let's say, where the label says you take one pill and somebody took 20 pills and had something bad happen, that would be categorized under an adverse drug event. And then even a further subset of an adverse drug event is what's called an adverse drug reaction. And so that's an adverse drug event that occurs when the drug is being used exactly as intended, right? And so this kind of speaks to the biological part of an adverse drug event, of something that could be expected. So, like a common side effect, for instance, of taking antibiotics might be diarrhea or something along those lines, right? So it's something that may be expected, may not be expected, but it happens when the drug is taken as it's supposed to be taken, as opposed to some kind of medical error or human error. So it's important to kind of distinguish those two things. And I think to kind of drive the point even further, I tend to like Venn diagrams and like to see these visuals. And I think it really helps me understand. So if we start with the universe of unfavorable and unintended occurrences, so this is our definition of adverse event, a subset of those are going to be caused by the treatment. And so that's the blue circle here. And then if we add in the element of medication or human errors, that's the black circle here. And so when we have the intersection between these circles, different portions of them correspond to the terms we covered. So the very basic one, adverse event, as we said, that's the red circle. That's kind of just any unfavorable or unintended occurrence that happened. And the ones that are caused by treatment, so the intersection of those two circles, as we said, is the adverse drug event. Now, it's important to show that the adverse drug event, it has a part of it that is, caused by medication errors, and a part of it that's not. A part that's kind of within the bounds of the black circle, and a part that's not. The part that's not within the bounds of the black circle, those are the adverse drug reactions, and that kind of is the distinction there. And I think, at least for me, it kind of helps visualize the differences between those terms. And finally, just to bring it home even further, is just to show it from how a clinician might interpret it in a flowchart form. And so if a physician, let's say, gives a drug to a patient, and the patient does experience some kind of event, the question is, was that event harmful to the patient? If the answer is yes, that's kind of categorized as an adverse drug event. And then those adverse drug events can be further subdivided, whether it falls within this gray box, which represents medication error. If it falls within the gray box, it's what they call a preventable adverse drug event. And if not, if it's something that's more biologic, that's not a result of human error or medication error, that's an adverse drug reaction, and that's the definition there. So I think that really makes it clear to me as well. There's also a kind of a flow where it didn't cause harm to the patient. They talk about intercepting potential events and things like that, but that's kind of beyond the scope of what we're covering here. So with that, we'll get into a little bit of the epidemiology. Adverse drug events and reactions, they really do represent a huge burden to the healthcare system. I can tell you they can account for 1 to 5% of all hospital admissions. And if we look at the elderly, the estimate is closer to 10%. And so those numbers really kind of, they really jumped out at me, because if you think about all hospital admissions, just anytime somebody is admitted to the hospital, 1 to 5% of the time, that person is admitted because of an adverse drug event, right? And so that's a much higher proportion than I would have expected, just having to estimate it myself. And up to 10% in the elderly, and that actually makes sense as well too, right? Because elderly people tend to have chronic diseases and things like that, and they tend to just be on more therapy to begin with. And so if you're taking more drugs, there's always just a higher potential, a higher opportunity, right, for a bad thing to happen from one of those drugs. And that's why I think the number is even higher there. We're seeing a 2 to 10-fold increase. Some estimates, and for some risk strata, it can even go as high as 50%. If you're taking just a whole lot of drugs, there's a higher potential. So those estimates really do have a wide range depending on the population you're talking about. Adverse drug events can also occur frequently during hospitalization itself. And there was one study in Italy that was published that made an estimate of around 6%. So regardless, you're in the hospital, regardless of why you're in the hospital, while you're in the hospital during your stay, about 6% of patients experience an adverse drug event during their stay. So that to me was also a number that's a lot bigger than I thought it would. And you think about the costs and the burden and the potential harm associated with those types of things. Adverse drug events also tend to occur more for inpatients than outpatients, and that's because inpatients are treated more for acute disease, and again, are on more therapies, and which again increases the potential for something bad to happen. And the final point I want to make on this side is that medication errors themselves can occur in many scenarios. And there was actually a super interesting study published in the UK recently, this year, that kind of quantified this in some of the various settings. So looking whether you're talking about the prescribing phase, transitioning phase, dispensing, administration, and monitoring phase, those are all potential areas for adverse drug events to happen. And in this study, head and shoulders above the rest was the administration phase when they quantified when the errors actually occurred. So that gives a little bit about just kind of the background, the epidemiology, kind of just setting the stage for what the burden is on the healthcare system for some of these adverse drug events. If we get into reporting, here I think we can kind of divide the clinical world into two parts. We have the experimental part, which are clinical trials, right? And then we have kind of the real world part, right? Which is kind of outside that experimental part, is just what we're experiencing here in the real world. So with clinical trials, adverse event reporting is, I mean, it's great, because you have a very clearly defined protocol that has very clear instructions, almost like a recipe that you follow if an adverse event occurs. So if an adverse event occurs, here are the things you do. And it's very laid out, very clearly step by step. So as a result, in clinical trials, adverse event reporting is very systematically performed, protocol driven, and very standardized, right? Because there's a very clear set of instructions. So if an adverse event happens at this site versus that site, there's a very clear standardized way that it's going to be reported and accounted for. And so that's kind of the ideal setting for reporting adverse events. But we don't live in the experimental world, right? We live in the real world. So adverse drug reporting in the real world is actually quite the opposite. So first of all, it's not automatically done. Secondly, it is usually spontaneous and anecdotal. So it's usually like, doc, I took this drug you prescribed, but I'm really getting headaches. And whether the doc responds to that or not is a totally different thing. But it's usually that kind of reporting, and there's not a pathway for it to be officially documented some way. Additionally, it's not mandated or required. So neither the patient nor the physician have to report these adverse events. And so as a result, we see a lot of underreporting, right? That's assumed with some of these adverse drug events simply because it's not mandated. And finally, I wanted to mention that the FDA here in the United States has a federal adverse event reporting system, but it has a lot of limitations. And actually, if you go to the website, there's a huge disclaimer saying that you shouldn't use this reporting system to estimate any kind of prevalence or incidence of any of these adverse events, and just very, very clearly laid out with that caveat. So there is very much an unmet need here in the real world and in real world data to understand what's going on with these adverse drug events. And then finally, to kind of set the stage more for Vic and talk about kind of the problem here, we find that in electronic health records, some adverse drug events are more detectable than others, and that's usually marked by seriousness. So just to give you an example, for example, let's say that I'm taking a therapy, and the adverse event I have because of this therapy is, let's say, liver failure, something very serious. So if that were the case, the physician more than likely is going to work up a liver failure. They're going to do a full workup for liver failure. There's going to be a diagnosis code. They're going to formally diagnose me with liver failure and prescribe the drugs and everything. So there will be a very, very clear record as a diagnosis code that, I took the drug on this date and I experienced liver failure 3 days later. And we can make the association that way. And it's going to be very systematically collected in the structured data. But let's say instead of liver failure, my side effect was something like headache, or I feel a little lightheaded from time to time, or something along those lines. More often than not, the physician is not going to do a full workup for a headache, right? There's not going to be a diagnosis code that says the patient had a headache. More often than not, if it is noted, it will be in the clinical notes. So the physician might write in the notes, patient came in taking the therapy I prescribed and is complaining about a headache, or something along those lines. And then there's other situations where it might not be in the clinical notes at all. Maybe the physician just says, you have a headache, well, just go take some ibuprofen. You can get it at any pharmacy, and may not prescribe ibuprofen, may not even note it. It just might be something anecdotal that was never recorded at all. And so that's kind of the problem we're experiencing with electronic health records. So I think that really talks more to the trade-off between the structured and the unstructured part. And finally, just to set the stage for Vic, is that I think that a reliable and systematic way to mine and analyze the unstructured clinical notes to detect some of these adverse drug events would be really, really highly valued. And so I think that's where we'll come in with using Spark NLP to be able to detect some of these adverse drug events. So with that, I'll turn it over to Vic.
Thanks, Lawrence. I'm Vic Kumar. I'm a data scientist at OmniHealth. And now that Lawrence has given a background in epidemiology of adverse drug events, we can go on and talk about how we mine adverse drug events from unstructured clinical data using Spark NLP for healthcare. So here at OmniHealth, we have an end-to-end framework for extracting adverse drug events from clinical text. And in our framework, we have 4 steps, and those are shown here. And in the next few slides, I'm going to go into more detail about how these 4 steps are performed. So the first step that we use is de-identification. And de-identification is important for unstructured clinical data because it prevents any protected health information, or PHI, from appearing in the result. You probably know that there's a series of laws here in the United States called HIPAA, and those prevent any sharing of protected health information between non-privileged entities. So de-identification is important to comply with privacy laws all around the world. So here we have an example input to this stage of the pipeline might be, as listed here, Mrs. Wilson is a 76-year-old female whose cough is probably due to lisinopril. And an example output of this step would be the same text in which the PHI has been replaced with tags. So you see patient is an aged female whose cough is probably due to lisinopril. So the name of the model that we used is called DID NER subentity glove, and that's a pre-trained pipeline trained by Spark NLP, John Snow Labs, and their Spark NLP for Healthcare package. So on their webpage, nlp.johnsnowlabs.com, you'll find documentation for this model and all other pre-trained models trained by John Snow Labs. So on the documentation page, you'll see a description of the model. Here you see entities that it predicts, and you can see some of the entities below. They correspond to the 23 protected health information fields outlined in HIPAA. And there's not shown here is also the accuracy metrics of the model. So it's a very useful documentation page for finding out more about their pre-trained models and pipelines. So here's the result, intermediate result of the de-identification step. This is an actual de-identified note. It's an excerpt of a physical exam for a patient. And you can see that the elements of protected health information have been replaced by tags. So you see the date tag up in the top, and you see the hospital has been replaced, and also the name of the physician has been replaced by tags. And so now that we've completed the de-identification step, the next step in the pipeline would be to do the main part, which is the adverse drug event extraction. So really there's 3 substeps to this part of the pipeline. And the first step is you need to identify the adverse drug event that occurred. The second part is you need to identify the drug that occurred. And then the third part is you need to see if those 2 things are related in a causal manner, as Lawrence outlined earlier. So you might have as an example input, the output of the previous step, patient is an aged female whose cough is probably due to lisinopril. And then in the output of this step, you would see cough extracted as the adverse drug event, lisinopril extracted as a drug, and then another field not shown here that would indicate either a 0 or a 1, whether or not they're related or not. So if we go to the next slide, here's an excerpt from another example. It's an infectious disease progress note, and you can see in the orange outline, this patient is on cycle 1 azacitidine, now admitted with febrile neutropenia and mild intermittent diarrhea. Azacitidine is a chemotherapy drug, and it is known to cause these 2 things, neutropenia and diarrhea, listed here. So if you go to the dark green box, there you'll see the drug has been extracted in this azacitidine. If you go to the light green box, you'll see the adverse drug event has been extracted, and that's the mild intermittent diarrhea. And then the blue box is a relationship field, and that is a 1, indicating that the drug and the adverse drug event in this case are related. So that's how the adverse drug event step of the framework works to identify adverse drug events in unstructured clinical text. So at first glance, you might think that now that we've identified the adverse drug events, we can call it a day and finish here. But in reality, when working with unstructured clinical data, there's some safety measures we have to take to prevent certain unwanted occurrences from happening. So there's two main unwanted occurrences. The first one is if you had, say you had a patient, his name was Mr. Cough, and he came in and his last name was misclassified as an adverse drug event. And therefore, in the result, you would have his PHI leaked in the result. A second occurrence we want to avoid is, say you had two patients on lisinopril. One was identified in the note as having cough, and the second one was identified in the note as being coughing. So there, the text strings for the adverse drug event are different, even though they're referring to the same diagnosis, and therefore it would be impossible to aggregate between those two events. So to solve those two problems, we use something called code resolution. These pre-trained models and pipelines are provided, again, by Spark NLP for healthcare, and they're a way of mapping the raw text in the adverse drug event stage to actual clinical codes found in RX norm for the drugs, and then ICD-10 for the diagnoses. RX norm is just a standardized code set for that has a code for each drug commonly prescribed. And International Classification of Diseases version 10 is a systematic way of identifying diagnoses and assigning them codes. So here we have it as an example from the output from the previous stage, which is cough. And as an example output of the ICD-10 resolution would be cough being mapped to its ICD-10 code, which is R05, and then having the corresponding description for that code, which in this case happens to be also cough, but is denoted here as a capital letter to note that it's not from the text, but it's actually the description of the clinical code provided by the World Health Organization. So if you go on to the next slide, we did the same thing for the drug. You have, as an input, you have the drug lisinopril, which is a raw text from the note. And as the output, you have 29046, which is the RX norm code for lisinopril. And again, lisinopril is capitalized to denote that it's from the RX norm and not from the text. So we did, again, here are two examples. And here in the orange outline, you'll see the ICD-10, the adverse drug event entities mapped to their corresponding ICD-10 codes. So here, if you look at the text above the orange box, you'll see age, year-old with a history of certain diseases, now admitted with febrile neutropenia and mild intermittent diarrhea. In this case, the entity that they're identifying is neutropenia, and that's mapped to its code of D70. And the drug is acetylcysteine, and it's mapped to its RX norm code of 1251. And then in the below example, you'll see the first sentence says therapeutic opioid-induced constipation. So there's an adverse drug event caused by opioids. And then in the orange box below, you'll see constipation mapped to its ICD-10 code, K59, and then opiates mapped to its RX norm code of 114189. Okay, so now that we've done the first three steps, we're nearing the end of our pipeline. And in this step, we do some post-processing. And the purpose of this step is just to reduce false positive matches, increase the accuracy of our pipeline. And there's three main things we do here at OmniHealth. And those are, first, we want to filter distances that are greater than a certain threshold. By distance, I mean, it's sort of like a confidence level of how well the match is. We want to filter out distances that are too high that might indicate a poor match. And I'll talk about that in the next slide a little bit more. We also want to filter out certain code categories like obstetric and pregnancy codes, which are associated with false positive matches for ICD-10. We found when we were doing this exercise that a lot of codes are mapped to obstetric and pregnancy codes. So we wanted to filter those out to remove those false matches. And sort of related to that is we want to filter out impossible diagnoses. So if you have a child and he's mapped to a code of adult onset rheumatoid arthritis, we want to filter out that because we know that's an impossible diagnosis, even though it was indicated using that natural language processing. So on the next slide, we do, here's an example of which we found the distance thresholds for RX norm codes and ICD-10 codes. So on the left, you'll see our overlapping histogram for RX norm code matches. The orange bars are the incorrect matches, and you can see that they tend to have a higher distance than the blue bars, which are incorrect matches. And so we picked n equals 4 as a threshold on which to filter out the poor matches. And then on the right is the same graph for ICD-10 codes, and we picked n equals 5 as a threshold there to reduce the false positive matches. Okay, so we've gone through and described our pipeline, and now we can share some results. Here's an example of the adverse drug events found for codeine, which is a drug used to treat pain. And we're working on a study right now, and we use maybe a couple 100,000 notes that we extracted adverse drug events from. And these are all the matches for codeine. And you'll see on there some nausea, some vomiting codes. You'll see allergies and rash reactions. You'll see hallucinations. And if you were to look at the product labeling, you would see that this corresponds pretty well to the adverse drug events indicated in the product labeling for codeine. We also have another example on the next slide. And here is an example that will not be in the final results of our study because it is not a prescribed drug, but it is a substance, alcohol. And this is just to show that some of the adverse drug events for alcohol are pretty specific, and they're picked up by the framework. And you can see pancreatitis is there, cirrhosis of the liver is there, toxic effect of a substance. And so there you can see that the framework picks up various adverse drug events for different substances, and it does so with good accuracy. So we've gone through and described the framework that we use for adverse drug events, and we've given some results. And with that, we're going to stop there and take any questions. We thank you for your time, and Lawrence and I hope you have a good rest of the summit.